Hello everyone and welcome to this week's mini video on prokaryotic replication. In this presentation, I'm going to use a model made of candy which you can replicate at home to practice. To begin, a prokaryote is a single-celled organism that lacks a distinct nucleus. One of the most common examples of prokaryotes is bacteria, such as E. coli. A characteristic of a living prokaryote is its ability to replicate its DNA to survive. Unlike eukaryotes, prokaryotes only have one origin of replication, which is where DNA begins to unwind and replicate. The two strands separate from the double-stranded DNA at the replication fork. Our example only has one replication fork, thus we will be demonstrating unilateral replication instead of bilateral replication, which has two replication forks. As DNA unwinds, the anti-parallel nature of two DNA strands will have one strand that runs 5' to 3' and one that runs in the 3' to 5' direction. The strand that runs 5' to 3' will be synthesized continuously and will be called the leading strand. The strand that runs 3' to 5' will be synthesized discontinuously and will be called the lagging strand. In our model, the bottom strand will be the leading strand and the top strand will be the lagging strand. Let's begin talking about the different proteins and enzymes that play a vital role in DNA replication. The enzyme gyrase is a topoisomerase that relieves the strand of any supercoiling before the replication fork. Helicase breaks the hydrogen bonds that exist between the bases of the nucleotide strands, but it does not initiate the unwinding of the double-stranded DNA. That is the job of initiator proteins. Now we can start to see DNA synthesis occur. Because the lagging strand replicates discontinuously, it forms short segments of DNA known as Okazaki fragments that will later be linked together to create a continuous strand. Next, we will see an enzyme called primase, which synthesizes short segments of RNA nucleotides called primers to provide a 3' OH group to DNA polymerases to attach nucleotides. The proteins known as single-stranded binding proteins attach to the exposed single-strand DNA to protect the nucleotide chains and prevent secondary structures like hairprints from forming. The lifesavers on the model are called DNA polymerase III, and their job is to synthesize DNA by adding new nucleotides to the 3' end of a growing DNA strand. The enzyme DNA polymerase I was the first polymerase to be discovered, and its job is to synthesize DNA and correct errors. It does so by removing primers and replacing them with new nucleotides. Now, what are the consequences of replication if DNA polymerase I can no longer perform its exonuclease function? The primers will not be removed and the gaps between the Okazaki fragments will not be filled. Thus, we would not be able to produce a continuous and functional strand of DNA, and the prokaryote would not be able to replicate. Last but certainly not least, we have the enzyme ligase, which seals the breaks in the sugar-phosphate backbone by catalyzing the formation of phosphodiester bonds without adding more nucleotides to the strain. Now it's your turn to try making a prokaryotic DNA replication model at home. Thank you for listening, and I hope you enjoyed this presentation on prokaryotic DNA replication. Here are my references.